So let's talk about the ways that these molecules can get through. The best way that you can actually transfer something over a bilayer is called passive transport. So when things undergo passive transport, it's just the molecules crossing over. There's no exchange of energy because we don't want to waste energy. That might be a hard thing for us to grasp as humans, but think of the biological world where a lot of organisms, they might only have one meal in a day. They do not want to waste their energy. If they waste their energy and they get attacked by a predator, they won't have enough energy to escape or get away. Or if you don't have enough energy to chase down and catch prey, you're not going to get a meal. And remember, biology is very binary. You live or you die. So passive transport is an amazing thing. Even for us as humans, we'd rather do passive transport because this is an exchange of materials that is not going to cost us any energy at all. We don't have to waste any energy to do that. That is a really good situation. You'll also see that word homeostasis has finally popped up. Homeostasis is the balance that our body is trying to maintain everything at. So if you are at homeostasis, or if your body is maintaining homeostasis, it means that your body is at balance. You don't have any excess or a lack of anything in particular. If there is, your body is going to maintain that. So if you have an excess of water, your cells will release more water. If you're lacking water, your cells will absorb or they will take in more water. That is just to maintain this homeostasis. So depending on what your cell is going to do, you're going to be able to determine what comes in and out of the cell. If you have to stay alive, cell must exchange materials, food waste, and its environment. Yada yada. We talked of all that stuff. A couple of molecules that we know about that can move freely are going to be water, oxygen, and carbon dioxide. That is typically going to happen through passive transport. Okay, there's so much of this, and this is almost always there. There's no reason for us to have to waste energy to get that in and out. These guys have kind of a free pass to get in and out whenever they want. So that's kind of like the principals here at our school. Principals can enter any room that they want. They have a key for every single door. They can pop in and out whenever they want. And that makes sense because they should be able to do that. A couple of larger molecules, things like the proteins and carbohydrates, well, that's where it gets a little bit tricky. You can't just let them in or out willy nilly. That's a little bit harder to do that. That's because our cell membrane is semi permeable. So it depends on where you are and what time it is. Sometimes things cannot get through. So that's kind of like having a bouncer, or if I had like a security guard at my door. Only some things are allowed to get into the room at a time. So if there's nobody in my room, if I'm on a prep, and if this is a normal year, I don't care. Anyone can come into the room, have your lunch, not a big deal. But if I'm teaching a class, well, I don't want just random people coming into the class. I'm going to have something at the door. In this case, I'm just going to shut the door. But if I had a security guard, the security guard would make sure no one gets in, or a bouncer. No one's going to get in until the class is available for someone else. Another type of passive transport, it doesn't use any energy, is called the diffusion. It's where small molecules can pass through the membrane and they just spread into the membrane. So it's the movement of molecules from an area of high concentration to low concentration. Definitely take note of this, high concentration to low concentration, because this is a theme that we're going to see in the cellular respiration and maintaining homeostasis. If you ever have something in high concentration, you will always move that to low concentration. And that's just something that exists in nature, right? If you have a waterfall, you have a waterfall because there's a high concentration of water at the top of the fall. That water is going to pour off the edge of whatever cliff it's at as a waterfall to a place of low concentration. Or if you have a balloon and you squeeze the balloon, so you filled up balloon, you squeezed it, you have created high pressure inside of that balloon. There's a high concentration of air or gas or water molecules in that balloon. Because the pressure is really high, or because the concentration of molecules are really high, those molecules want to get out where the concentration of molecules are fewer. And if you squeeze hard enough, that balloon will burst. And as you notice, the balloon does not keep its shape. All the stuff on the inside gets out. So it gets from a high concentration to a low concentration. Diffusion works like that as well. We call this difference in the concentration a concentration gradient. And we look for these in all parts of our body. In fact, our body's going to create concentration gradients to aid in hopefully passive transportation or passive diffusion. Another example of diffusion that you might be able to relate to better is the use of scented products. So let's say I brought a bottle of perfume to the classroom, and at the front right of the room where the periodic table is, I squirted a tiny bit of that perfume into the class. And those of you that sit closer to the front right of the room, close to the periodic table, you might smell that perfume in a couple of seconds. But those of you sitting at the back by the eye wash station or the door, you're not really going to smell anything for a while. But through diffusion, that spray was sprayed in high concentration. There's lots of perfume in that one corner of the room. It is going to move to an area of low concentration, which is the rest of the air in the classroom. So slowly, those scent molecules are going to spread out throughout the entire room. And even you sitting all the way in the back next to the eye wash station are going to smell that perfume when that gas gets a chance to get there because it's trying to equalize itself from that high concentration when it was sprayed out to the low concentration, which is the rest of the class. Even the classroom is trying to maintain this balance, not homeostasis because the classroom itself isn't alive, but it's trying to maintain a balance where every single part of the classroom has the exact same amount of perfume molecules. So it's going to spread them out to get that balance. Just an image of what that looks like. You can see it with water as well. If I put a drop of food coloring in water, you'll notice that that water kind of or that food coloring clumps in one area. And if I let it sit, it slowly starts to spread itself out. And if we wait for the duration of a class or maybe a couple hours, you'll notice that the entire water has now taken up that food coloring. That's because that highly concentrated food coloring has diffused into various parts of that medium. So this is driven by what's called kinetic energy. This is something that every single molecule possesses. We can't see this because obviously molecules are really small, but it just means that the molecules are vibrating a lot. Okay, so they're literally just vibing. Like the molecules just they sit, they vibrate back and forth, and the more energy you put into a system, the more they bounce around. So because of this kinetic energy, molecules are always in motion, and that's going to bounce that other molecule, the thing that you're trying to fuse, from place to place to place. It's in a random pattern, but it will spread them away from each other in either a liquid or a gas. And in gas, it happens very quickly. In a liquid, it happens slightly slower, but you can witness it. The rate of diffusion, it's going to depend on a couple of factors. One of the main ones is temperature. If you increase the temperature, it means that you're increasing the kinetic energy. So that's why if you're dancing, for example, you get hot as you dance more, right? Or if you're working out, the more you do, the hotter you get. Same thing with the molecules. If we increase the temperature, the molecules are going to move faster because they're trying to release more of that energy. They have more kinetic energy and they're going to bounce around more. And if they're bouncing around more, the molecules that we're trying to diffuse will also be bounced around more and they'll fill up that space much more quickly. I'll show you a demonstration in class. Depending on when this is posted, you might have seen it already, but a demonstration of how food dye will diffuse across a body of water, either at room temperature or if the water was warmer, or if the water was warmer and it had some additional kinetic energy added to the system in some other way. You also have to watch out for the size of the molecules. If they're a lot larger, then there's less of these collisions that are happening in the water. So it's better to have smaller molecules and higher temperature instead. Again, just to break that down, molecules diffuse faster at higher temperatures than at lower temperatures. Smaller molecules will diffuse faster than larger molecules. Most of the short distance transport in our cells is going to be through diffusion because it's very short distance. All you need is to create a concentration gradient. If you have that, you're going to move from high concentration to low concentration. Okay, diffusion will always happen down that concentration gradient. It will always be from high concentration to low concentration. So a high concentration to low. When I spray that perfume, it's never going to cluster in one very small little ball of space. That doesn't make any sense. It's going to spread out from high concentration and spread out to the low concentration all throughout the room. When molecules disperse evenly, there's no longer any diffusion because there's no longer that concentration gradient. The whole purpose is to find balance. That's what homeostasis is. So when you finally get over that concentration gradient, you have completed the diffusion, you have balanced out that concentration gradient. you've balanced everything out so you don't have a concentration gradient anymore. So that's what the goal is. And that's why the body creates these gradients if it ever wants to do some sort of passive transport. The fusion will eventually cause a concentration of the molecules to be the same all throughout. So going back to the food dye example, put food coloring in water, you'll see a few clumps of that coloring in the water, but once the diffusion has happened, all of the water will be the exact same color because the diffusion has made the concentration the same everywhere. When the concentration of a molecule in a substance is the same throughout any space, we call that equilibrium. If it happens in a biological setting, we call that homeostasis. And our body is always trying to maintain that homeostasis. There's another type of passive transport that can happen as well. This is specifically with water molecules if you want to get water to go from one area to another. So if you want water molecules to go from an area of high water concentration to an area of low water concentration, again, that concentration gradient is applied. We call this osmosis. So water can move across a cell membrane from an area of high to low concentration. And this is going to be through that process called osmosis. Just like anything else, it's going to move from a high concentration to low. It goes down the concentration gradient. Just an image of what this looks like. So this is actually what an actual YouTube looks like. So not the media sharing platform, but a YouTube is a U-shaped tube. And let's say we had a selectively permeable membrane in the middle. If we had, say, a low concentration of solute, not water, low concentration of solute, and a high concentration of solute, what that means is that if there is a low concentration of solute, you have a higher concentration of water on the left-hand side. That makes sense, right? Because look, both halves of this YouTube are at the exact same level, but the right half has more of these green dots, which means it has less of this blue space. On the left half of the YouTube, you can see there's more blue space, which means there is more water. So what's going to happen is the water is actually going to balance itself out. It's going to cross over this membrane in the middle through osmosis, and it's going to fill up the right side so that the concentration on both sides are the same. So yeah, the YouTube is going to force the water through, and it's actually going to balance out where the right half is going to be taller than the left half. And you're correct. There is less water in the left half than there is in the right half. But we don't care about the amount of water. We care about the concentration. The concentration on the left is going to be identical to the concentration on the right. Okay, so be careful with the words that we use because yes, things go from high concentration to low concentration. But as you can see, the water went from low concentration of solute to high concentration of solute. I'll flip my words around here. That's because the thing that I'm talking about, the movement of water, is not indicated in these pieces. I'm talking about the actual concentration of solutes. So make sure when you're assessing a situation, you want to talk about the thing that's actually moving. The water is going from an area of high concentration. There is more water molecules on the left side, or there are more water molecules on the left side. There are less on the right side. So that's why the water moves over. The sugar concentration allows for that difference. So that's why we mention it. But the water is still going from high concentration to low concentration. Balances everything out. So osmosis will always work in transferring water over, and that is going to be manipulated by the amount of solutes that you have. So the amount of sugars that you have, and salts in particular, are going to be very big with this. So solutes are dissolved substances in a solution. The cytoplasm is mostly water, and it's got a couple of these solutes in there. And it depends. If you have more solutes in the cell than you have outside of the cell, chances are your cell is going to bring more water in. If you have more solutes on the outside than you do on the inside, your cell is going to let more water out. Because no two molecules can occupy the same space, so that's both the solute and the water molecules. Don't forget that water is also a chemical. It has different molecules. They don't necessarily have to be in the same volume, so that can change the actual flow of the water back and forth. But some solutions will have a whole bunch of different solutes in them. And if that's the case, they're going to have fewer water molecules than those that have fewer solutes in them. So water is always going to move from low solute concentration. Again, be careful with my words here. Water is going to move from low solute concentration to high solute concentration. That's because the water is crossing over from a higher concentration of water to a lower concentration of water. And that's actually going to balance out both. The concentration of water is going to balance out on both sides of the gradient, and the concentration of solute is going to balance out on both sides of the gradient as well. So the net direction of osmosis depends on the relative concentration of both sides, and the ability of a molecule to diffuse through a cell is going to be dependent on a couple of factors. One of them is the size of the actual molecule itself. Since most molecules diffuse through the pores of the membrane, smaller molecules are able to also diffuse much more rapidly. So what that means is the membrane has these teeny tiny holes that the water molecules can get through. But if you have something that's smaller than a water molecule, why can't that get through as well? Molecules above a certain size, however, cannot go through these pores, through these holes. Another thing is a chemical composition that we're looking at. So certain types of molecules, there is a Comic Sans font here. I'm sorry about that. Certain types of molecules will diffuse through a membrane faster than others, and that depends on the chemical that you've got. Cell membrane is more permeable to some classes of compounds because it might be made up of different types of proteins than it is to a different type of protein that's there. So fat-soluble compounds, for example, will go through a membrane much more quickly than another compound of the same size because fats and fats work really well together. Whereas if you had a fat and, say, a carb, the carb is going to have a harder time getting through the fat layer. So it has a bit of a challenge doing that. The other thing to watch out for is the charge. Okay, so this is where chemistry comes into play. Chemistry is just small charges. It's really small electricity on different elements. It's difficult for a charged particle to pass through a membrane than it is for an uncharged particle. And that's the effect of having equal or opposite charges. So if you have a charged particle, you probably have other charged particles in your membrane. They're going to repel it, make it a lot harder for that charged particle to go through. Or other things are going to be attracted to the charged particle before it gets into the cell, so it gets slowed down from actually getting in there. Also depends on the type of membrane that you have. So your membrane of the cell might be more permeable than another membrane, so that's going to influence how this works as well. And that's going to bring us, since we're talking about the solutes and the charges, to the tonicity of a solution. And this is the strength of a solution in relationship to osmosis, and that's going to help us identify which way the water is going to go in or out of the cell. Normally, we have a situation that's isotonic. This is where the solute concentration of both solutions is pretty much equal. It's in balance. You can have a hypotonic solution. This is where the salt concentration of one solution is less than the other. You can also have a hypertonic solution. Hyper means more, so this is where the relative concentration of a solute is greater than in the other solution. So a couple of pictures of what that looks like. The middle image is when you have an isotonic solution. So if water comes in, water comes out. It's in balance. That's what the cell should be. If you have a hypotonic solution, that means that water is going to be flooding into the actual cell. When that happens, if there's too much water getting into the cell, you're going to get a lysed cell. It's going to pop just like a balloon, and that's going to kill the cell. If you have a hypertonic solution, it means that there is more of the solute outside of the shell. So the water is going to leave the actual cell. It's going to shrivel up the cell and make it less useful. It's going to make it a little bit less pliable. So it's also not the greatest situation, but that can happen sometimes. Okay, so let's say, for example, you just went to McDonald's and you got some fries. You just ate a whole bunch of fries, didn't have any water. Well, now your solution outside of your cells is full of salt. That means the concentration of solute is much greater, so that your cells are going to release that water to balance that out and not make your blood full of salt instead. So your cells will get shriveled up, not the greatest solution, so you probably want to drink some water in that case. If you are quenched and you fill your body up with water, you got too much water in your blood, well, then your blood doesn't really want that much water, so your cells are like, no problem, I got this. They'll suck up that water for you. But if you have too much water, some of your cells will actually start to burst because there is too much water in there. And as you can imagine, that is catastrophic because as soon as the cell bursts, that water is back into the bloodstream, and then another cell is going to start sucking that up. So you don't want to have a hypotonic solution. That's essentially how you drown yourself from the inside out. So in tonicity, we can see that actually in plants as well. Plants do this. They use hypotonic to absorb more water. We call this a turgor pressure. And this is why plants will maintain an erect position. That's why they're tall. And this is why they wilt when they are thirsty. So when they have a lot of water in them, it allows more water to be flowing through the cells themselves. They have big vacuoles that hold them upright, and so they can use this turgor pressure to hold them upright. There are solutions that cause cells to shrink, which are hypertonic solutions. Red blood cells, for example, if you place them in a salt solution above 0.9%, so that means if you have like a mostly water solution, there's like barely anything, like barely 1% of that solution is salt, your blood cells will actually shrink and shrivel up. As you can see here, you got a couple of cells that don't look very round anymore. We call that crenation, and that's not a good thing. We don't want your blood to have 1% salt. You want to have much less than that. Next up, we move into what's called facilitated diffusion. Facilitated diffusion, this is a passive transport across a membrane that still doesn't require any type of energy use. And we've got the diffusion and osmosis that are going to do this for us. Most molecules can't cross by simple diffusion, and we might need some help through things like carrier proteins. So those are those guys that are going to open the doors for us to let us in. So just so you can see what that looks like, in this case, we got passive transport when we're facilitating that diffusion. Simple diffusion means a molecule just goes through the membrane. If you have a channel protein, you might only allow certain molecules to get through depending on what size. So if this yellow dot was bigger, it's not going to fit. It's not going to passively transport over. You could also have a carrier protein of some sort. Carrier protein needs to trap that molecule and allow it to get into or out of the cell. There's a different type of situation called active transport where you have to spend energy. We don't want to do this, but there are some things that we just have to spend energy to actually work. That can be a carrier protein, where the carrier protein will accept the molecule, but in order to actually allow the molecule to leave, it has to pay. And it has to pay through the currency our body uses called energy. So that's like if you got to my front door and the only way I'm allowing you into my room is if you give me a $20 bill. That's how I let you into the room. That is active transport. Okay, you don't want to spend your money to get into the classroom. We don't want to spend our energy to get in or out of a cell, but sometimes that happens. In this case, we have to actively transport. We want to avoid this if possible because we're spending energy, but if we have to spend energy, if we have to do this, we have to do this. Okay, so carrier proteins are going to change shape based on how they are attached to whatever a molecule is in them. The change shape that allows a molecule to cross the membrane, and the use of these carrier proteins for diffusion, we call that facilitated diffusion. This is going to help the substances move in or out of the cell, depending on that concentration gradient. A good example of this is glucose. Glucose type of sugar. So a lot of our cells depend on glucose for their energy needs. Facilitated diffusion is going to allow passively that transport of the glucose in or out. So there's no energy input. Gates are going to open and close depending on the conditions and the environments in or out of the cell. There are three different kinds of stimuli that are going to open those gates, and that's going to stretch the cell membrane, or it's going to be an electrical signal, or it's going to be a chemical that's in the cytosol or the outer environment of the cell. Any of those three things can influence how that facilitated diffusion happens, right? If you stretch, you open the canal a little bit more so that cells can get through a little bit easily. If you change a charge or if you get a signal, you might be able to attract or force something in a little bit more easily, or you might have a chemical that does that, opening up some of the spaces in your cell. The opposite of that is this active transport. In some cases, we need to move things up a concentration gradient, so from low to high, which shouldn't happen. So if we want to actually do that, we have to put some effort into it. So when I spray that perfume in my class, if I don't want it to spread around the room and I want to get it back in the bottle, I have to catch it somehow. So I'll probably spray it in a box. I got to close the box up, not allow any of that to escape. In order to do that, like there's a lot of thinking, there's a lot of actual work that I have to do. So in that case, I'm keeping things from a low concentration and bringing them into an area of high concentration. That's not how physics should work, but we can do this by spending energy. Okay, so active transport, we've got adenosine triphosphate. There's your adenosine, there's your phosphate group. It will break into energy when you break some of those bonds. And when you break those bonds, you're breaking one of those phosphate bonds. So you end up with adenosine diphosphate and phosphorus. That's how you get the energy. One of the last things we're going to talk about is endocytosis and exocytosis. Some of our molecules are very complex proteins. They're too large to cross over a membrane, so these have to cross by bulk transport. In bulk transport, the large molecules, things like food and other substances, they're packaged into a membrane, and those sacs, those are called vessels, are moved into and across the membrane. So that's if, let's say, it's like a Trojan horse. If I want you to get into my classroom, but the door is closed or something like that, maybe you'll hide yourself on the bottom of a cart and someone from the office brings a cart into my room, and all of a sudden, ta-da, you're in the classroom. Awesome. Another way that you can do that is if I covered my door with, say, I don't know, let's say I covered it with like some sort of like bubble mixture, the kind of mixture you use to blow bubbles. If you want to get into my classroom, you're going to get that bubble mixture all over you. But that's one way that you can do it. You wrap yourself in a bubble and you're in. Okay, so bulk transport, you wrap yourself up in something. That way you can deliver it to places that they need to go. Large molecules, food, and other substances, they can be packaged in this way and sent into a membrane. So a couple of ways that we see this. Endocytosis is when we are leaving the cell. Pardon me. Endocytosis is when you're entering the cell. One way you can do this is through phagocytosis, where you have a solid particle. It ends up going to the plasma membrane. The plasma membrane will wrap itself around the food particle and it will escort it into the cell. You can also have penocytosis. Penocytosis is similar, except we're talking about the fluids instead. So if you want a fluid to get in, same thing, it gets onto that plasma membrane. The plasma membrane will wrap it up and escort it into the actual cell. You can also have a situation where you have a receptor mediated situation. It could be endo or penocytosis, where you need to have the actual receptor react with some sort of molecule that fits into the receptor. That is going to allow a specific protein to bring in the product you're looking for. So in this case over here, I've got this pit and I've got these three receptors. Once those three star molecules get sitting on those receptors, the receptors know that they are full. They're going to wrap themselves into a circle, and that is going to come into my cell from there. Okay, so we call that endocytosis, exocytosis, penocytosis, and phagocytosis. Just a combination of all of them. Endocytosis, the membrane is going to fold into a pouch that encloses the particles. The pouch pinches off, and that goes to make a food vacuole that goes into your cell. Okay, so the vacuole will then fuse with other organelles, things like lysosomes, to be digested and break off into pieces. Both peno and phagocytosis are different types of endocytosis. Peno is called cell drinking because you're bringing some fluids into the cell. The solids or fluids outside of the membrane can be brought in to the cytoplasm using penocytosis. At the same time, we got phagocytosis. If I want a larger food particle to get into my cell instead of a liquid, we'll call that cell eating. Phagocytosis comes in, the lysosome will break down that particle, and now we have that food into the cell, and whatever nutrients it had are into the cell. The food vessel will then fuse with that lysosome, as mentioned, and it will break down, and we'll be able to use it. In other places, you got things like white blood cells, in particular in your blood. These are going to help you destroy bacteria and other unwanted cells. It uses phagocytosis as this mechanism of action. So the white blood cells, they eat bacteria and they destroy them from the inside. We don't typically use anything in the bacterium, though. It just gets flushed into your kidneys and then into your bladder, and you just pee it out. So what happens in your body is these white blood cells will sometimes even engulf old blood cells. This is a regular occurrence. When it does that, it will break down the old cells and it will reduce them and eliminate them from your body if they can't reuse those pieces. Remember, we want new cells all the time, including your blood cells, so they have to be replenished on a regular basis. Exocytosis is the opposite of endocytosis, so that's leaving the cell. Usually we do that with waste and other cell products. So if I make a protein in the cell, I might use exocytosis to get it out. If I have waste in the cell, obviously I want to get it out of the cell. I'll use exocytosis to do that. So quick image of that. Endocytosis on the right. Particle comes in, packaged inwards. Exocytosis, package comes to the edge, and it is released into the extracellular fluid. Then we get into ATP. ATP is adenosine triphosphate, as we mentioned before. That is the currency we're going to be using for our cells. So when cells need energy, they're going to spend ATP. The more demand for ATP means your body needs to produce more ATP, so you need to make sure your body has fuel to do that. The amount of ATP is usually recycled in a very small amount because you have ADP and P, which can be obviously like mixed together pretty easily. But you still need to replenish that ATP because not all of it, we don't have a mechanism of action for reusing all of it. So I don't need you to worry about memorizing the cycle or anything like that. Again, I'm using that word memorize because I don't think it's really that useful, but you can see the ATP group here. You've got the adenine, you have the ribose sugar that is connected to the triphosphate group, hence adenosine triphosphate. It is broken down to adenosine, diphosphate, the one phosphate is left over. Usually we can remake those, right? Because it's a pretty easy fix. We use some sort of food or some sort of sunlight to do that. Our bodies aren't as good at doing that, so we don't do it as much, but we can. Energy from ATP when you create ADP plus P is just about enough for most biological reactions. Sometimes you need a bit more. As mentioned, you got a base, adenine, you got a sugar, ribose, and then those three phosphate groups. It is a high energy compound because a phosphate group is very easily removed, but phosphate groups hold a lot of energy. So when you break them off, you release that energy for your cells. That ATP can be used for a whole bunch of things like chemical work. That's going to allow your body to do different chemical activities. It's good for transport. We need to move things from place to place. So you can use energy to pump substances across a membrane, specifically with active transport. You can also do mechanical transport to move things like the cilia and the flagella or chromosomes so that they get to where they need to get to. And that's going to bring us to the end of our notes for this section. So thanks, guys, for sticking with me on the cells and cell bodies. I keep apologizing. It's a pretty dry unit when we go through it at the start of the year, but hopefully this is a good intro into the course, and then we'll get into a couple of activities and try getting into our body systems shortly. Anyways, hope you're having a good day. Thanks for watching.